Hello everyone, myself Dr. Ashish Jain. I am Assistant Professor in Microbiology Department of Srimati CHM College, Ulhasnagar, which is affiliated to University of Mumbai. Today I am going to present an outline of course on gene mutation and repair. The assignment is given to me as a basic requirement of 5th refresher course in ICT, which is being organized by UGC-HRDC Indore Center. I would like to thank Director of HRDC Indore, Dr. Namrata Sharma Madam, and Course Coordinator, Dr. Shaligram Prajapat and Dr. Surend Malviya sir for giving me opportunity to develop course content on this topic. So friends, the topic of today is gene mutation and repair. So now we move to the next slide. So the course content is divided into four parts. First part will deal with the introduction of gene mutation. Second is type of gene mutations. Third is detection of mutations. And fourth one is repair of mutation. So this is the first part of the course, introduction of gene mutation. So first we have to understand what is mutation and what is the normal condition or the normal function of a gene. So first, the normal function of gene is when a gene produce normal product, means the protein which is produced by the gene is a normal protein. But when there is a mutation, which is a defect in the DNA base pair or a chromosome, so the gene product may be defective or there may be abnormality in the protein or no protein at all. So ultimately mutation affect the protein which is the product of a gene. So mutation start from alteration in a base pair and it result into abnormal gene product or abnormal protein. So this abnormal protein will affect the normal metabolism of the cell. The second topic of our syllabus is types of gene mutation. So gene mutations broadly divided into two categories, uh, base pair substitutions. Under this, transition and transversion are two subcategories. So let's see what are the transition and transversion. So here we can see the transition mutation where a AT base pair, this is converted into GC base pair. So when purine pyrimidine will alter into another purine pyrimidine, it is called transition. Second category of base pair substitution is transversion mutation. So in transversion mutation, here CG will convert into GC. So pyrimidine purine will convert into purine pyrimidine. So this type of mutation is transversion mutation. Some other uh, consequences of gene mutations are the missense mutation. So where altered amino acid is result. Second is nonsense mutations when coding region will be converted into a stop codon. Next category is the neutral mutation where there is no significant alteration in the function of protein because the amino acid that is synthesized due to mutation has the same property of the wild type amino acid. Last category of the base pair substitution is the silent mutation. So in silent mutation, although there is a change in the base pair, but there is no alteration in the amino acid, means both base pairs will code for the same amino acids. Next category of the mutation is the frame shift mutation. This mutation is either deletion or addition of the nucleotides. So here you can see uh, if one base pair is incorporated in the DNA, then the whole reading frame will be changed where the base pair is incorporated. So frame shift mutations has two categories: base pair insertion and base pair deletions. Now the mutation, types of mutation. So there are basically two categories of mutagens. First is the physical mutation, in which the radiations are there. Some ionizing radiations and non-ionizing radiations are categorized into physical mutagens, and chemical mutagens basically divided into base modifying agents, base analogues, and intercalating agents. So this slide represents the intercalating agent. So here profilin, flavin and acridine orange are the example of intercalating agents. So these intercalating agent will incorporate in the between the base pairs in the DNA, and they distort the morphology of DNA, and they cause mutations by additions and deletions. Under the physical mutagens, X-rays, UV rays, gamma rays are main agents. So in these, X-rays and gamma rays are more severe, than they cause drastic changes in the DNA. But ultraviolet radiations are responsible, they are mild radiations, so they can cause the pyrimidine dimers. So here we can see the thymine dimers are formed in the adjacent base pair of a DNA strand, and due to this covalent bond which is formed between two adjacent pyrimidine, DNA polymerase cannot synthesize DNA beyond the point where these pyrimidine dimers are formed. Under the chemical mutagens, base modifying agents are there. So here we can see nitrous acid as a base modifying agent, which convert uh, cytosine into uracil, so that the Cg base pair will convert into Ta base pair, and at the same time on adenine it will convert adenine into hypoxanthine. So At to Gc base pair is the result. Other base modifying agents are the hydroxylamine that convert cytosine into hydroxyl methyl cytosine. So Cg base pair will convert into Ta base pair. Next base modifying agent is the methyl methane sulfonate, which is an alkylating agent, and it converts guanine into O6 methyl guanine, so that Gc base pair will change into At base pair. Intercalating agents we have already discussed. Now, detection of mutation. So this is the third topic of our syllabus. So how we will detect the mutations? So some mutations can be detected by the visible observations, like the eye color of the Drosophila. So the wild type, we can see the variety of eye color in the Drosophila. So first one is the wild type and other 8 are the mutants. So some mutations can be detected just only by the visible observation. Nutritional mutants, nutritional mutations are detected by the replica plate technique where uh, prototrophs are discriminated from the auxotrophic mutants. So auxotrophs are those organisms that require particular nutrients for their growth, while the prototrophs are the wild type. So this type of mutations can be detected by the replica plate technique. Next is the Ames test. Ames test generally be used for the screening of potential mutagens. So generally what happens, mutagenic compounds are also carcinogens. So this Ames test determine whether a chemical which is under investigation have the carcinogenic potential or not. So this is a complex experiment which is performed on the histidine auxotrophic Salmonella typhimurium, and due to the presence of the chemical, if the auxotroph convert into prototrophs, then the compound under investigation may be the carcinogenic, and if there is negative results, then the compound may be not harmful. So this Ames test is used for the screening of potential mutagens which may be the carcinogenic. The last topic of the course is repair of mutations. So mutations are generally reverted into the normal condition by the automatic processes which are inbuilt in the cell. So here we will discuss some of the crucial procedures. So these are different repair systems. So we will discuss them one by one. So DNA polymerase proofreading activity is present in the DNA polymerase enzyme. This is actually 3' to 5' activity. So due to this activity, most of the mutations or most of the mismatches are recovered automatically. Pyrimidine dimers are repaired by the photoreactivation, that is the light repair mechanism. Most of the mutations that are related to the alkylation are repaired by the O6 methyl guanine methyltransferase enzyme. Base excision repair system uh, is conducted by the glycosylase enzyme, where the abnormal bases are replaced with the normal one. Nucleotide excision repair is the complicated repair system, and this repair system is conducted by the 4 proteins. Uh, Here we can see UVR-AB, UVR-C, and D. So these 4 proteins together uh, perform a repair mechanism, and at the last DNA polymerase and DNA ligase enzyme will operate. Methyl directed mismatch repair system. This is conducted by 3 different gene products, MUT-S, MUT-H, and MUT-L. So this type of repair system also used to correct mismatches, and ultimately DNA polymerase 1 and DNA ligase fill the gap. The most severe repair system is the translesion DNA synthesis. This is also called SOS repair system. This is conducted by the LAX-A and RAC-A protein, and this is little bit complicated repair system. So there is an event of inhibition or the activation of the genes which are involved in the repair mechanism. So in this course we will study in detail such repair systems. At last, in the summary, the effect of mutations. What are the different effects of the mutations? So first thing is mutations are essential for the evolution because they increase genetic variation and the potential for individual to differ. So the main reason of evolutions may be the mutations, random mutations. Most of the mutations are neutral because they does not affect the normal functioning of the cell. So there are many mutations in a cell, but these mutations does not affect their normal functioning. Some mutations which are beneficial, uh, they are incorporated permanently in the cell through the natural selection. So mutations are also beneficial, and due to these mutations, organism get adopted in the extreme environmental conditions. But of course, some mutations are harmful and they cause severe changes in the metabolism, including genetic disorders or cancers. So, we will discuss all these things under this course. So, thank you for participating in this course. This is a presentation uh, by me, Dr. Ashish Jain. Thanks again for your patience listening.